Right, so this is the sort of second set of lectures on bionics. What we're going to cover today are animal experiments of one kind or another. Animals in this sense meaning not humans. So we're sort of doing everything. So we can pick up, the results and relate them to humans, as we can see. And clearly there is a little bit of a gap from those. For those of you that were here for the first section, obviously you know all this, but in case you wanted to email me, there's my email address. You want to find out all the dirt on me, it's probably on the Wikipedia site, and find all the truth, then it will clearly be on my own website. The course, what we have already covered previously in October and November time, were the first three parts, looking at general introduction, and then looking at artificial organs in terms of human parts, and then arms and legs. What we're going to be covering today in this first presentation, the animal experiments, relating them where we can to humans, as you'll see, but specifically looking at experiments that have occurred with non-humans. And then we're going to look at neural interfaces. I did originally say sensory and motor, but I thought I would pull the motor. It sort of relates in here and the later two. So I've kept this more for sensory. So we'll be looking at artificial retinas to an extent, and the cochlear implants, things like that. So it's more the neural type of things. And then we'll get into deep brain stimulation. The main uses of deep brain stimulation are more for Parkinson's disease and epilepsy. Those are the things we're not going to cover here. So I'll look at mainly what it's about, and then look at more of the, not fun things, that's not really, but the more wide varying neurological issues where deep brain stimulation is used. So this is pushing electrodes right into the brain for therapeutic purposes. Parkinson's disease and epilepsy, then we'll cover more next time round, more how it's used for that, where it is used mostly. So the first one, we're covering now animal experiments. And clearly the whole reason for experimenting, there's all sorts of ethical viewpoints. I'm not going to go into the ethics, whether this is good or bad. Please don't take it that I'm saying this is a good thing to do. This is just what's happened. As scientists, we can't always agree with what's gone on in the past, but there's the scientific results from it. So we take the results and go from there. So I'm not going to discuss the ethical aspects of it. And not really looking at the chemical side of things, and not the sort of external sticking things onto animals and stuff like that, or cosmetic. Behavioural, we'll see. Humans are animals, so there is a link, but in the neurological sense, it can be questionable. Tipu Aziz, who is a surgeon I work with on a regular basis, unless it's in a monkey, he's not interested. So experiments with rats and things, it just dismisses because the rat brain is too different to the human brain. Him, as a surgeon, that's his choice. But clearly other experiments on rats and so on, which for some reason rats are not particularly popular, so people can experiment on them more easily. In England we have all these animal rights, people love animals for some reason, so give you some example, for the experiments we've done, it takes us about 9 months or more to get ethical approval to experiment on anything to do with rats. To experiment on humans takes us about 3 months to get ethical approval. So it puts it in balance in England, the relative importance of animals and humans. And again, more interested here on the neural aspect, how the brains are functioning. And in Prague, of course, we have to start with Pavlov. He's got a metro station named after him, and so on, so you have to start with him. He was quite a guy. We're looking now over 100 years ago, but he really did move the field on with his experiments. As we will see, I mean, he won the Nobel Prize in 1904, so obviously what he did was of some significance. But the sort of things he did, if we look back on them now, were really quite horrific. I mean, people, when they passed through the metro station, if they knew what this guy was doing, they would probably, while they're eating their McDonald's there, he would bring it back up again. So things he did took out portions of the digestive system from animals, mostly dogs, but he was working on anything he could get hold of, really. He cut nerve bundles to see what that would stop happening or cause happening, and then finding the effects. He put valves between the digestive organs and out to an external pouch, so he could see what was actually coming out. So he rerouted the digestive method. The animal would eat, but it wouldn't actually go into the animal's stomach. It would go out somewhere different. In Reading, we have a pub called Pavlov's Dog. For some reason, you know, again, Pavlov is known throughout the world. He actually had lots of dogs and lots of different animals. He started work which linked in with what Freud and his researchers were looking at, more in humans. But he looked at the effects of stress and pain in what was called transmarginal inhibition, TMI, and how the body, if it gets overwhelming stress or pain, how it starts to shut down different aspects, partly as a defensive mechanism and so on. But it was really looking at how quickly some dogs, in this case, shut down parts of their body in response to pain, and how other dogs, it took longer. And where's the difference there? You know, again, potentially looking with people's subject. And I'm not going to go into women and men, and women and feminism. I'm not going to go into that argument. But clearly, some people can put up with a lot more stress and pain than other people. And why is that? What's the difference? Is there something, say, neurological, but is it a physical thing in the nervous system or what? So, I mean, what his research showed, that all types responded in the same sort of way. So parts of the body started shutting down in the same sort of way, but how quickly that happened was different. Some groups seemed to be a lot more quickly, a little bit of stress, shut down. Others could put up with a lot of stress and pain and so on. And he came up with the conclusion that it was due to different types of nervous system, that some had stronger nervous systems and some weaker nervous systems and so on. The popular belief, Pavlov, what he did was to ring the bell and then give the dog food, and ring the bell, give the dog food, and then when he rang the bell, the dog started salivating. So it was linking this noise of the bell to food. The bell brought about behavioural responses, neurological responses in the animal. So that's what he was looking at. Which we get, I mentioned McDonald's, you get the same, show the colour red, oh, I need something to eat, all that sort of thing. So now we know, particularly with marketing, lots and lots of features there. And sometimes psychiatrists are trying to figure out what is the trigger mechanism that brings about a problem for a person, and can we remove that trigger mechanism. So it's really going back to the work that Pavlov was doing. Some sort of trigger responds, causes a particular response. But he didn't just use a bell, he used whistles, tuning forks, looking as well at the different physical noises, sounds, images and so on that could cause a particular response. His work, again, this is where it links in with Freud. Carl Jung picked up on it and was looking in different types of animals in terms of introverted and extroverted temperaments. So he switched it more to the psychological side, the psychiatric viewpoint, not so much on the physical, how the nerves and the neural makeup was, but more from the outside, the psychological. And showing his results, he felt, showed that introverted persons, people who are quiet and don't say so much and so on, were much more sensitive to stimuli. In other words, didn't put up with the pain and stress so much as extroverted people who go around, hey, hello, how are you, that sort of thing. For some reason, seemed, from what Jung said, seemed to be better at dealing with pain and stress. Now, there's all sorts of questions there. Does that mean if you are an introverted person and you started to become more of an extrovert, you would be able to put up with pain and stress? So, you know, could you learn to put up with it more? It raises all sorts of interesting questions. Carl, some of what Jung did was questionable with regard to humans, and I'm not going to go into, I'm looking at the scientific side of things. William Sargent as well was starting to look at different ways of manipulating because of that. And his work went into brainwashing and implantation and all sorts of things, based on the same approach. Delgado is one hell of I can't go through this without mentioning the guy. If you did a Google search on Delgado, he comes back with all sorts of weird stuff. But he was, in terms of animal experimentation and the whole field of neural monitoring and manipulation, incredible guy. He, way ahead of his time, I have to say. He developed what he called the stymoceiver, and this was a radio signal which he said stimulated brainwaves. So it was either EEG or more likely through an implant into the brain. The implant was the thing that he actually used. Again, more animal experimentation initially. And he, in terms of stimulation of different parts of the brain, produced a variety of effects. And he again worked with a variety of animals mainly, and produced, so he claimed, pleasant sensations, elation, deep, thoughtful concentration, odd feelings, super relaxation, visions. So this is pushing electrodes in the brain to bring about all sorts of different results dependent on where the brain was stimulated. Now clearly some of these you could perhaps infer from animals, but when it comes to coloured visions, and it can only be through human experimentation that you get those things. And this is where you see, if you read what's out there, Google and read, you get all sorts of different impressions and feelings of the work that he did. He has a mixed response. One well-reported incident is he was Spanish originally, but he moved to the US. He stepped into the ring with a bull, and the bull was charging him. The bull had one of these implants in its brain. It charged Delgado, and he had a remote control button, which at one point he pressed, and the bull just stopped charging him. So, I mean, he said it caused the bull to lose its aggressive instinct. The point was it stopped charging him, and that was from him pressing a button. Clearly, you instantly leap from that happening in the brain of a bull to could you do the same in the brain of a human? And in what sense would that make? You know, if I put an implant in your brain, and whatever it is, you'll be okay, it might be aggressive, whatever you're trying to do, I press a button and you stop. Or I press a button and you do something. You know, what are the possibilities there? He said although that was very visual, and he did it partly for people could see, look, that's what's, you know, it's there, and it was a well-reported experiment, that's what happened, and so on. He said his experiment with a female chimpanzee was more significant. Again, this chimpanzee had a sternoceiba, and he was looking at when what he called spindles, certain neural activity more deep in the brain, and producing an aversive reaction. So it was, you know, exactly what he tried, different things. So when a particular thought, he used the implant to bring about bad feelings in Paddy's brain, or if you like, pain signals, whatever you like to look at it as. Oh, and the feedback was within the brain. So because there were negative signals produced when the monkey thought in a certain way, so the monkey started not to think in a certain way. So it's a sort of negative training, if you like, but within a feedback loop. Delgado, different reports, said he was Spanish, he went to America and eventually disappeared into sort of the military research within America. And a lot of his work didn't appear in technical papers, and so it's very, very difficult to judge. Again, this thing with the bull, there it was, you know, before the days of videoing, there were a lot of reports on it. But when he disappeared into the American military, you can only guess. I mean, you can see what area he was working in, but no reports came out at that time. So there we go. So he went for the money and continued his research. I'm picking, as we go through, some sort of fun things that are perhaps not so well scientifically reported. Obviously Pavlov's work was very well scientifically reported. I mean, here's one, it's as much of a fun thing, but it gives you an idea the sort of things that can occur. So this is from the University of Tokyo, electrodes into the antennae of a cockroach, and signals were just then sent in remotely. You know, it's great having the little lights to show when the cockroach is going right, left, and so on, backwards, forwards. So it was remote control of the cockroach through electrical signals. We'll see the same again with rats and so on. And there's similar work with moths into the antennae with chemical signals. But I just picked that as one example. There's quite a few examples of that. This is a nice piece of work. Musa Valdi, Northwestern University, US at Chicago. Got a, as we'll see, little two-wheeled robot. We'll see a video of this in a second. I've got some sound on videos later on, but this one, the sound is not that important, so I'll talk over it. Now, what you're going to see, as we'll see what a lamprey is. They've taken out part of the lamprey brain. They can keep it in solution for about 24 hours. And essentially it's linked up to a robot body for simple things. Yeah?
Sorry, what was it,
a lamprey, which you will see, it actually goes back. It's like an eel, like a snaky fish. I don't know what the word is in eel in Czech. I don't know. It's fine. But we'll see. It's a small one, and in Lake Michigan, which this is Chicago, Lake Michigan, there's a lot of these lampreys, so you just get the thing. And again, nobody cares about lampreys, so experiment, they can just get ahead, do the things, and so it goes on. So they can keep but the main thing here is there's part of the brain in the lamprey that simply aligns this eel in the water, so it keeps it the right way up. So where there is light coming in, it will make the lamprey go a certain way. And what they've done is take that part of the brain and link it up to a robot body. So light causes the robot to do something. So this is Mustard Development. He's a really nice guy. If you get a chance to listen to him or talk with him, then take it. So there's your lamprey, your little eel. So it's a very small thing, but it's quite an old thing from prehistory. It's not development. And here's a little robot, and as we'll see, so this is sort of keeping the lamprey brain in a solution. So it is sort of alive, but it's really just taking out part of the brain, kept there, dies within 24 hours, so it's not sort of pseudo-alive, I guess. And what we'll see, so in that little corral, they will then switch a light on, and with the response to the only control of the robot is via this lamprey brain. Now we'll see more of this type of an approach later on. But again, the biological brain is determining which direction the robot goes. So if there's a light, it goes towards the light. If there's not a light, it either goes away from the light or doesn't do anything. So it's a by choice, either this or that, and away it goes. But it's taking part of the biological function, the simple biological function of the brain, and using it to control a piece of technology. The extension from this would be other parts of the brain to do other things with a robot, and clearly with humans to take part of the brain and use that to do some particular functioning or whatever. So there we go, animal experimentation of a different kind. Along the same lines, but something more, and this will link in with probably next time round's work, Miguel Nicolelis, North Carolina. He took a monkey with the brain functioning in the monkey and linked it up to a robot arm, so that the robot arm mimicked the motions of the monkey's arm. And the monkey could see what it was doing, and therefore you got some sort of feedback visually. Now this, you have to remember this is in the US, so the words used here, you have to think of them being as though it's an American saying, oh, it was an amazing sight to see the robot in my lap move. That's me pretending to be an American. Knowing that it was being driven by signals by a monkey brain at Duke. So it was looking, rather than just doing it in the lab, fine, this was doing it over a distance, and the monkey could see and made the links between what it was doing and what the robot arm was doing. And this is an important point. It was as if the monkey had a 600-mile long virtual arm. So you've got a connection from the monkey brain to this robot arm, and visually back the other way. This is my comment. There was no direct feedback, it was visual feedback. I have to say, I was with Miguel Nicolelis, I know him a little bit, and I was on a panel a year ago with him, and there was a question from the audience about science fiction. How does science fiction affect what you do? And he would not answer the question. He would not go there. Well, no, I'm not going to answer the question. Which I guess is because of the funding and so on. He doesn't want to even go there in case the people who give him the money stop funding him. But it's disappointing, really. I mean, as scientists, we can be affected by science fiction if we want. Why not? So it was disappointing. Clearly, there are aspects of science fiction that can excite, but a nice piece of research all the same. John Chapin, I was saying about the other linking up to the brain to control movement and things like that. He's been doing this sort of thing with rats for some time, based in New York, and his main line of work, well, there's two two parts to the experiment. The one we're going to see first in terms of a video, movement signals, well, signals transmitted from a computer into the rat's brain. It has, as you can see, a little, this is a radio receiver on its back, and I can't probably catch this, but electrodes pushed into a couple of positions in his brain, so they can send signals from the computer into the rat's brain. He describes it as one electrode stimulating the feel-good centre. It's positive to say it's a feel-good centre. Two other electrodes activate things to do with left movement and right movement, with the idea that at a particular point where the rat is, if they can send a signal, this feel-good signal, to get the rat to want to move to the right or want to move to the left, then that's what the rat will do. He calls them stimulus cues. So here, this is John Chapin. This is actually pinched from a Korean TV program, but it's quite a good coverage. But I presume that there's not too many of you fluent in Korean, which I'm certain about two words is all I know. So there's John Chapin. So here's the rat, and you'll see straight away the electrodes, which they're just clipping onto there. So the electrode permanently positioned in the rat's brain. There's the backpack you can see as well. So he seems quite a happy chappy, the rat anyway. And this is the maze setup they've got. So you can see, I think at this point, then there's the computer, which they can just click right, left, so the rat will head in the particular direction they wanted, or they can change the direction and off he goes. Go to the right, the rat goes to the right, go to the left, and the rat goes to the left. Reasons for doing it, all the scientists are always looking for reasons apart from pure research. And here, the idea is the rat has got a camera on its backpack as well, so they can see where it's going, and to get the rat to look for bombs or dangerous weapons or find people, I guess even, where it could be a dangerous area. So there's the idea they can remote control the rat to go into dangerous areas where humans might be injured or destroyed, so who cares about the rat? But there we go, you get an idea. And again, the point is with this, okay, this is what we can do with a rat, we can get it to go to the right or left or whatever. Could we do the same sort of thing with a human? Well, obviously we can. The human brain is more complex, and if we can do this with a rat, then we could do that with a human plus a lot more. How the signals appear in the brain, here we're looking at direct signals going into the pleasure zone, so or they could be negatively into the pain zone, but it's direct signals to cause the rat, in this case, it's a person, to do this or do that. Now, with marketing, you're getting a much more external. It's perfectly legal, it's okay to show certain stimulus cues to make you buy this product or to go down that aisle or to buy. So that's okay. Here we go more directly into the brain. Hey, should we be doing this or should we not? There's something not so okay.
And I'm wondering if the rat will be on an edge of some, I don't know, something. And can you force by sending the signals to do the rat fall down, to go against the fear? Or is the control of the rat stronger than the control? The area of the brain they go into here is more the planning area. So it's not just the motor response area. So it is more at the stage where the planning, the rat, if you like, is having a conscious thought about this is what I'm going to do. So I don't know the answer. I don't have the same direct answer, but it is at an earlier stage. So I would say it is more likely that the rat thinks it's doing what it wants to do. It's not like, you know, I want to go left, but it's actually going right against, no, I want to go this way, it's having to go the other way.
It's more a case of, yeah, this is a good idea, I'm going to do that. If it had natural fears, because in the maze there's not, it doesn't matter one way or the other, unless there's some reward or something like that. But if it had a natural fear, so it would be interesting. Yeah, whether it, and I'm sure John, he's the guy, to ask John, send him a, he's a nice guy, so I'm sure he would be happy to respond. So I don't know exactly that.
I have a question as well. So you said that they connected the one electrode to the pleasure area, and then both areas, one referring to the left and one to the right. And what was the basis that first there was a stimuli from the left-right area, and if he let's say, complied to go left or right, and it was reinforced like in Pavlov's signals by the pleasure system, or it was simultaneously?
It's come to a certain point.
Yeah.
I mean, it might initially want to go to the left, but then they would send a pleasure signal and go to the right. So it associated pleasure with, at a pertinent point, if I go right here, it's a pleasurable experience. If I go left, then
so, do they have to condition first those responses, or they just connected it and it's automatically like plug and play system?
I think it is a plug and play.
Plug and play, okay.
I wonder what would happen if you send both left and right signals.
You've got to go and work with John Jacob and try it. Again, he is quite a fun guy. He looks quite serious there, but he's quite a fun guy, so I'm sure he probably has tried those things, so it wouldn't surprise me. Again, relating it to how the human brain works is important. Sort of separate experiment, again using rats. Into the planning area, he took a group of 6 rats, got them, they were learned to pull a lever down to get a little sweet liquid, which they seemed to, a sugary liquid. So every time they pulled the lever, they got this sweet liquid. He took signals from the implant in the brain so that when they planned to pull the lever, so you could best figure it, then signals were used to cause the sweet liquid to appear. So effectively, they didn't have to actually carry out the plan, the action. So as long as they thought about pulling the lever, that was fine. The liquid would be released. And it's interesting, out of the 6 rats, 4 of them learned pretty quickly they didn't actually have to. So they stopped physically pushing the lever. So they thought about, they planned, I'm going to push the lever, but they didn't actually do it. 2 of them still kept pushing the lever for some reason, the stupid end of the rats, I guess. Still kept doing it even though they didn't need to, didn't figure that out. But again, you think for humans, if you think about doing this, doing that, you don't have to do it, so you don't do it. You get in the reward or whatever it is without actually doing it. We'll see experiments again with rats and in terms of stimulation of the brain, lots of, unfortunately, quite a bit in Germany through the years. I don't know why I'm going to say any more about that politically. I'm getting myself into all sorts of trouble. In 1870, Hitzig and Fritz stimulated the brains of dogs, and this was picking up, look, these areas in the dog's brain are to do with motor functions. So stimulating the dog, and the leg would move, and arms and things like that. So remote controlling bits of the body moving around. And Bartholow, in a few years, demonstrated the same in humans. So stimulate parts of the human brain when the arm moves around, and so on and so forth. Krauss did a whole mapping of the human brain using this, so people were undergoing brain surgery, and whilst they were having surgery, so he would be pushing electrodes in and saying, hey, look at what happens when, there we go. Ethical, but you learn, I guess. So Hess into electrical stimulation in the 1930s, so you can see where we're going politically in history, I guess, at that time, jolting patients' brains with shocks administered through tiny needles. So putting needles in the skull and putting electric shocks to see what different parts of the brain, what happened. Now, for us to and I mean, for some cat, well, it says cats, but I mean the reports are he did the same sort of thing for humans and so on. Putting into particular parts of the brain mild electrical stimulation and the just crazy running around making screeching noises. What was actually going on in the brain? Difficult to know.
But were they still anesthetized or not?
Pardon?
But they were still anesthetized or not?
I don't think.
And despite the anesthesia, they were going berserk?
Going berserk. I don't think here that they were not. I mean, anaesthetised. We will see. I mean, for anaesthetics for the brain, it can be just local anaesthetic to overcome the pain. So it's not anaesthetics in sending the thing out. It's just over. So there's nothing from the pain of putting the electrodes in, which is the same in deep brain stimulation. There is a little bit of anaesthetic used, but not the patient's awake and everything like that. Well, so I can see what you mean, the berserk and that. We start to get, say, fun. Penfield experimented with brain stimulation. Again, patients undergoing surgery. One thing, this is interesting when we start to look at memory chips and things like that. What he discovered with patients that were awake, he stimulated different parts of the memory and the patient started remembering, recalling things. So he would put different currents in different parts of the long-term, short-term memory, particularly the long-term memory, and they would, I remember when I was a boy, or I remember 3 weeks ago, memories they couldn't remember having, and they would suddenly recall things from their childhood and so on and so forth. So the point that you can put electrical signals into the brain to stimulate memories is one important point from that. It then opens up thoughts of, well, if we can bring back memories, can we change the memories? Can we download memories? That kind of stuff. This I think is one of the most incredible pieces of research in terms of how it affects humans and so on. 1956, Olds. He started off with rats, but the experiment was as follows. Again, into the pleasure centre for rats that we've been looking at with Chapin. It's fairly well known. Fairly well known for monkeys, fairly well known for humans. So what he did for rats, with electrodes in the pleasure centre of the brain, simple button that the rat could push that gave the rat a choice. Either they could press the button which stimulated pleasure, or they could press a button which gave them food. And that was it, that was how they worked long term. He found that the rats, after a while, became so obsessed with the pleasure that they didn't bother with the food, to the extent that they starved to death. So here they have food that's there, and all they need to do is push the button, but they don't push the button because they can get pleasure if they push this other button, and they just go for pleasure. All right, these are rats, they're not going to... Well, he then did the same experiment with monkeys with exactly the same results. Exactly the same. The monkeys did it, starving themselves to death. Now, I'm not sure that people have done exactly, because of ethical issues, I guess, done exactly the same experiments on humans. But when you relate this, you think, oh, humans would never be so stupid. But when you look at drugs and so on, where people do, and even alcoholism and habits that are formed like that, that the person, they go for that pleasure fix, even though they're told you're going to die. If you, okay, it's not the same choice between food, but it is that you keep going for the fix, you can go for the fix, and you die as a result of it. And you do hear so many cases of that with alcoholism, with drugs and so on, a person dying because they're just so addicted to it and they won't give it up and stop and so on. Always cases coming along. Not in the academic world, of course. Right. Yeah, again, different pieces of research. Brain scans on monkeys. The desires, the planning, there's just a small number of cells, neural cells that are used, and then the plan to carry out particular actions and so on. And Caltech, they did a project with, more recently this is, for the monkey carrying out a simple video game, and linked up a computer program, a cursor and a screen and so on. So again, this was like the rats. In this case, the monkey thought about moving right, left, whatever, and the cursor automatically moved. And we'll see the same sort of thing next time round with some human experiments in terms of Philip Kennedy's work. As I said, Parkinson's disease we're going to deal with next time round with deep brain stimulation. We'll have partly because there's a lot of people involved in terms of Parkinson's deep brain. I picked up on Tipu Aziz, who's the guy that I work with. He's at Oxford, and they have quite a lot of monkey houses there, which get all sorts of problems from animal rights campaigners because their monkeys are being used for experimental purposes. And what with the monkeys, part of what he's about is bringing about Parkinson's disease in monkeys, and then using electrodes to switch off the symptoms. And we'll see next time around. I know at least one of you is researching in this area, but we'll see more of that next time. This is using deep brain stimulation to counteract the effect of Parkinson's disease. But Tipu does a lot of work with monkey brains to firstly bring about Parkinson's disease and then see where to position electrodes in the brain in order to counteract and what currents to apply. This, in terms of ethical questions, over 40,000 people who have Parkinson's disease have now benefited from this technique. So this is quite a number of people. It doesn't cost money, it's a medical treatment, but it's not like it's 2 people or something. 40,000 people. Parkinson's disease is a pretty horrific thing. So a lot of people can benefit from research on animals. In the UK, Tipu pioneered this thing. And it's Parkinson's disease, dystonia, which is like muscle locking. Is it part of Parkinson's disease or is it not? But it is alleviated. The electrodes are permanently planted, but next time round we're going to look at that in a lot more depth. I thought that we're looking at animal experimentation, and I thought, well, I'll conclude with this little bit that is something that I'm involved with. Some of you may know about it. If you do, fine. If you don't, well, fine. You will do in a second. It sort of fits, but it's a little bit different. So here we go. What we have is a robot body, like we saw with Musa Evaldi in Chicago. It's a technologically robot body with the sensors and wheels and so on. In this case, rather than using a lamprey brain, we've taken neurons from rat embryos and put them in a little dish. This is a multi-electrode array. So the electrodes are on the bottom of the dish, and we grow the neurons. So this time the neurons are alive and not just a short-term fix. And whereas with the Musa Evaldi work, maybe you didn't like this, I don't know. The Musa Evaldi work, it's really taking part of the brain from the lamprey and just using it as it is, some of the features. This is really looking at growing the brain and as the brain develops in its robot body, so it develops its own features and functions and habits, and learning is part of what it's about. So this is the multi-electrode array. These are called Potter rings, after Steve Potter that originally put them there. The neurons are squidged in there. The neurons are taken from rat embryos, and the earlier in the cycle, the better, so the neurons are more plastic. Typically there's 100,000 of them in the dish. The rings are to stop the brain culture dehydrating. After about 1 week of the neurons being put in there, they've connected up. Neurons like to connect up with each other, and we have like a two-dimensional brain in this dish, which we can send signals to and pick up signals from. And after about 10 days, we pole round and apply signals at different electrodes and see where there is a response. So let's say this is electrode 25, and we apply a pulse and we get a response from electrode 71 in a reasonable space of time. Then that is then connected to the robot body. So the sensors from the robot will stimulate the brain, and then the output from the brain will change the direction of the robot. So it is a bionic thing, it's biological and technological, but it's different in that the brain now, rather than still being in the creature or in the person, is being grown separately. So it's giving the brain a body, which is very Kafka-esque, to put the prime link in there. It's very much like Gregor's Metamorphosis and all that sort of. Re-embodiment, we believe to be essential. A lot of work is done just growing neurons together but not putting them in a body, and you can come up with all sorts of strange results because of that. Having some sort of input-output relationship, giving it some sort of input-output to the world is important. Where animals have developed, even humans, in a sensory-deprived state, then regions of the brain don't form suitable connections and so on. The person simply doesn't have the ability, or the animal doesn't have the ability. So in this case, you've got the culture of the brain growing live neurons. It lives in an incubator, 37 degrees centigrade, and the output from the brain has a meaningful result. And just to finish this first section, and then we'll take a break. So this little robot has a brain which is about 10 days old, and what it is supposed to do is detect the wall and turn right. And what it's not supposed to do is change its mind and crash into the wall. There we go. So this is just learning to get around at this stage. Again, it's just like a baby robot, really, mentally. The important point is that one can look at, this is the physical robot moving around, but you can look at how the brain develops. And what one can witness here, the brain is in the body for about 1 hour every day. And you can see over a period of time how the brain changes, purely through, in this case, through a process of heavy learning, habitual learning. Because it's doing this every day, the habit of doing it changes the neural pathways in the brain. So that's the robot, which you can see. Some of the time it doesn't succeed, some of the time it does. The important point is that every time it does get, like there, chunk, gets it right, that will strengthen the pathways in the brain that brought about that decision. Every time it does that, it becomes more likely to do it. That's the thing of habitual, doing something that is. So it's not like a reinforcement learning in the same way, but because that neural pathway occurs, and takes part in an action which the body takes part in, as it were, so it becomes more likely to happen next time, and more likely to happen, more likely to happen. So the same robot, same brain, after 2 months, and you'll see purely through the process of habitual learning what it's going to do. This one, in a sense, you'll see is probably quite boring though. There we go, off we go. Picks it up quite early. So remember, we saw it in its early life making a lot of mistakes. This one is probably a perfect student, I think. They're not all that good. Some of them do get it wrong even after a period of time, or occasionally make mistakes. But remember, this is a biological brain making this decision every time. It will get a sensory input and it's deciding, I'm going to turn right here. It could quite as easily say, no, I'm going to do something different. In which case we can have it turning left and all sorts of different things. But this is just choosing that every time. It might choose, no, it doesn't, as we saw earlier on, it might choose not to do that. So this is a different form of bionic animal sort of experimentation, but in a technological body. Trying to understand what memories are and how the plasticity of the brain affects things and so on and so forth.
How do you enforce your punishment?
Well, it doesn't actually get. We can, by applying chemicals, if we have a feeling in the brain where there are pathways that are doing things we want it to do, in this case making an appropriate decision, we can apply chemicals to that part of the brain so that the pathways are more likely to connect up and strengthen. Or apply chemicals to other parts of the brain where it's doing something we don't want it to do, that kill off the neurons or restrict the growth. So it's very, very less likely. So we can reinforce in that sense. In this one, it's not a form of reinforcement learning. It's purely this habitual learning. When you do something, and you do something, and you do something, every time you do that thing, whatever it happens to be, it becomes more likely that you're going to do it next time. This is what Donald Hebb spotted. Hebbian learning, one neuron fires, causes another neuron to fire, and in doing so, that strengthens the pathway between them, whether we like it or not. It is like habits. Every time you smoke a cigarette, it's the habit, you reinforce the fact that you're going to smoke more cigarettes. In a sense, because you do it, you're minded to do it again. Mind every time you have sex. There we go. So it's that habitual learning is what we can witness mostly on this one. And reinforcement in terms of electrical stimulation, we don't know where the pleasure zones are on this, so we can't really go.
Well, the thing I don't understand about this habitual learning is how do you make it to learn what you want? Like, why did you learn to turn right? Yeah. It just doesn't spin or something.
We cheat, really. Like I said, after 10 days, we will pole round here. And so the brain will have grown for about 10 days, just making connections, if you like, randomly. I mean, it won't be random, it's pseudo-randomly. It will have made connections and links. So we will then come along after 10 days and try each of the electrodes, apply a pulse here, and see, do we get a response after a few milliseconds somewhere else? We may not. If we get a response on a reasonably regular basis, which could be, say, 20% of the time, when we stimulate here, we get a ding-ding-ding-ding, and we get a response there.
So you just see how the brain is wired and then train the machine accordingly?
That's right, yeah. We just pick up, we have some sort of wiring that some of the time, 20% of the time maybe is sufficient. Maybe 10% would be sufficient. If we stimulate here, we get a response there. So we then connect those up to the robot. And then what you saw in the first robot, the 10 weeks one, 10 days one, sorry, was that 20% probably. I think it may be a bit better than 20% that one. But some of the time it was getting it right, what we wanted. Some of the time it wasn't. The point with that though is just doing that for the 2 months, you could see the difference. So we didn't then, once we connected up to those particular electrodes, we didn't change round, it was connected in that way. And then you can witness this habitual learning. I mean, I have to say, when I first studied artificial intelligence and told this is Hebbian learning, I thought, hey, what is this? But when you actually witness it in the neurons, and this is what actually happens. And it does, not only in the robot and the brain that you're working with, but then you see how that maps over into humans. And purely the fact that if you do something habitually, you become more likely to do it. Can be good or bad, it doesn't matter. Often it's bad, but it's best to stop doing what you're doing if you don't want to make it more of a habit. So, and you notice it when you're driving somewhere. You drive somewhere, and then in the end you realise I didn't even think what I was doing. I was just driving. I can't even remember what I was doing, because it's become so much of a habit to go there and turn left and turn right, and you end up not even thinking, or even going. If you go the same tram route every day, you've realised you're just doing it, and you didn't realise what you were doing. You didn't even think about it. You just did it because it's habit. It's this thing. So it is as though part of your brain is just scooting in and out the signals. This signal causes you to do a particular thing. You don't even think about it. I suppose a lot of the military training is towards that. You do it habitually. Ding, you do that. Dong, you do that. You don't question. You don't think. You just do it.